Okay. And with that, it's my pleasure to kick off this week's Prime Movers Lab technical webinar on Living Therapeutics. I'd like to start by welcoming our community here, our limited partners, our founders, extended PML family, and a special welcome to our new Growth Fund LPs. For those new to Prime Movers Lab, we are a venture firm focused on breakthrough scientific inventions that have the ability to transform billions of lives across energy, infrastructure, manufacturing, transportation, agriculture, and last but not least, human augmentation, which is where today's webinar sits. The purpose behind these webinars is to share some of the things that we get to encounter and learn about every day, particularly a lot of these amazing people with the rest of you all. We have a strong interest in making sure we bring up everybody. This is emblematic of our style, bringing together industry experts, founders, and investors to make the world a more interesting, exciting, better, all-around great place. And also to highlight some of the important work being done out there in the field by people like those on our call today. For those who don't know me, my name is Carly Anderson. I'm a technical partner here. My background is in chemical and biomolecular engineering. I learned very early on that I'm great at killing cells. I actually spent some of my PhD destroying them with plasma, leaned into that. And it's kind of the opposite of what the folks here are good at, which is making people better and manipulating cells to make us all healthier and have better lives. So with that, I'll kick it over to Caleb to introduce the panelist himself and what we're going to be talking about today.
Hello, everyone. It's an honor to be here today for this seminar. I am a joined Prime Movers Lab at the tail end of 2020. Unlike Carly, most of my PhD was spent on keeping the cells alive. I've been in the kind of living therapeutics area for quite some time. Living therapeutics, for those that don't know, were the original therapies humans had, and they're now in a little bit of a renaissance with some really interesting things. And I think we fast forward 50, 100 years, maybe only 10 years, people will look back and say, why were we treating diseases like cancer and Crohn's disease and various things with small molecules or things other than the living therapeutics? And today here we have some really amazing entrepreneurs. I've been honored to know all of them for quite some time. I think you will be incredibly impressed by their perspectives and the companies they are building. And with that, I will turn it over to Yug to introduce himself and what he's working on.
Thanks, Caleb. Hi, everyone. I'm Yug. I'm the co-founder and CEO of Phi Therapeutics, which is a microbiome company based in San Francisco, California, in the United States. Phi is working to change the way we treat chronic bacterial diseases, and we use technology that uses bacteriophages to modulate your microbiome. Briefly, we kill only the bad bacteria without hurting the good bacteria in your body. Now, this is really important because the microbiome, as we know, is known to be very, very important in human health. It keeps us healthy. Sometimes it makes us sick. And as we understand it more and more, we need a subtractive method to remove certain strains of bacteria without destroying the entire microbiome. We know how to add, and this technology allows us to subtract. I think we're all on the precipice and probably pioneers in the fourth great revolution in medicine. The first one was using extracts like St. John's wort to treat headaches from nature. The second was isolating small molecules, like isolating aspirin from the St. John's wort. And so we pioneered small molecules for most of the 20th century. More recently, we embarked on the third revolution, which was using biologics instead of small molecules. And now we're using whole cells, whether it's stem cell therapy, whether it's using bacteria, phages, or any engineered version of those. And like Caleb said, in a way, this is cyclical because we used to use whole cell therapies in the past, and now that has become the cutting edge. So my background is in molecular biology with a foundation in organic chemistry. I've been doing microbiome research for 10 years. And yeah, I think the potential of phages is great, and I'm looking forward to talking more about it.
Thank you very much. And Larry, perhaps you could give a little introduction of yourself.
Thanks, Caleb, and thanks for inviting me here today. Larry Weiss. I'm the Chief Scientific Officer and co-founder of Symbiome. We are a San Francisco-based skin microbiome and biotechnology firm. We've taken a bit of a different approach. What we've done is run 3 expeditions into the Amazon, built a deep map of what the ancestral human microbiome looked like 12,000 years ago. What's interesting is hunter-gatherers, which is how we spent most of our time on the planet, don't have any of the inflammatory diseases that we have today. They don't have cardiovascular disease, they don't have diabetes, they have no inflammatory disease of their skin, acne, eczema, rosacea, psoriasis don't exist. There are those of you who think, well, they have lived short lifespans, but their modal age of death is actually in the 70s. And we have lost about 80% of that diversity. So what we've done is built a map for this, and we've actually building isolates that restore the function that we used to have and turning these into skincare products, running clinical trials on these products. And the notion here is we spend a lot of effort in medicine and science treating disease and fixing broken stuff. But broken stuff was not our evolutionary legacy of health. So what we're looking at is how can we restore our biological resilience, which is the foundation of our health. Resilience in response to stress is health. My training, my background is in natural products chemistry, then in medicine, internal medicine, intensive care medicine, and anesthesia. And I have been working in the microbiome since the first data started to become available in the late 1900s, late part of the last century. The big message of the microbiome is everything is connected in ways that we did not previously appreciate. Most life on Earth is microbial. It's ancient. It nurtured us. It supports us. We are not the pinnacle of evolution. We are the spoiled and arrogant newcomers who are the beneficiaries of this $4 billion-year-old trust fund.
Thank you very much for that background. And a special welcome to Nogami-san, who is joining us from Japan at the wee hours of the morning. Thank you very much. Would you please give us a little background on yourself and what Metcela is doing?
Sure. Thanks for inviting me. My name is Ken Nogami from Metcela. Metcela is a Tokyo-based company launched in 2016. And we have been working on creating a new cell therapy for heart failure, chronic heart failure. Heart failure is known for really unmet medical needs, having more than 5 million patients in the US and more than 1 million patients in Japan. We are creating a cutting-edge cell therapy by using autologous, the patient's-derived cardiac fibroblast. We get special population and inject that back to the patient's own heart. I think we are the only company in the world having the tight relationship with a catheter company, a created proprietary catheter for the injection of the cells to the heart, so that we can do this treatment with a minimally invasive way and with a precise targeting of the cells, targeting by observing the heart in a 3D mapping system. And I think the cell therapy has been changing really rapidly with having new modalities for the diagnosis of the symptoms. Unless we have this kind of precise mapping, because the cells are not moving around in the heart, we couldn't have been achieved really high level of the efficacy in the animal models. As to the progress of the business, we are some weeks away from the phase 1 first in patients clinical trial. So we are really excited about that. And we have already achieved good efficacy in the rodents and the pigs. About myself, I am actually ex-investment banker at Morgan Stanley. So maybe I was really surprised to hear all the backgrounds the other people have, but I may have more business or financial background into the discussion. I'm really excited to be here. Thank you.
Thank you, and thank you all for joining us. A little housekeeping before we get into this. This is going to be a bit of a fireside chat. If you have questions, please feel free to put them in the chat at any time. We will try and fold them into the conversation. And all of the panelists, please feel free to ask each other questions. We'll run this till the end of the hour, and we'll try and reserve a couple minutes at the end for direct questions from the participants here. I'd like to just dive right into it, and how these living therapeutics are very different, and maybe for Larry or Yug initially, we're seeing these as alternatives to antibiotics in some ways. There is our battle with disease is exactly that. It is kind of a battle, a war, and with the antibiotic war, we've kind of clearly lost, because the bacteria evolve and penicillin did not. How do you see that playing out with phage therapeutics and microbiome therapeutics?
Yeah, I mean, that's, I think, the struggle of the age right now. I think none of this is needs underlining. I think everyone knows the gravity of the situation. And those who don't will soon be made aware of it, because the WHO says, and the UK health body says, that by 2030 or 2050, it's going to become a crisis. 10 million people will die of commonly procured bacterial infections that wouldn't otherwise. With antibiotics and the current model that we have, we are in a race with bacteria. And the race goes as follows. We spend a lot of time doing research and testing these products, as we should, for safety and efficacy. Takes about decades and billions, now tens of billions of dollars. The moment the drug hits the market, within a few years, if we're lucky, usually within a year, you start to see resistance. And then that resistance, given our modern lifestyle, people flying from one part of the world to another, our interconnectedness, that resistance really spreads very fast. So it is a race we are always losing. With phages, we have an ability or an opportunity to win this race. And the reason is that phages are the most diverse life form on the planet. It's estimated that there are, 10 to the 29 to 10 to the 30 bacteria on the planet. There are at least 10 phages for every bacteria on this planet. And they're so numerous that if you took every star in the universe, the number of phages on Earth, there are about 100 million to 1 billion phages for every star in the universe, not in our galaxy, in our universe. And so they are, to compare another cosmological term, they are the biological dark matter of our planet. They have unbelievable diversity. We know almost nothing about it, and yet they shape our planet in ways that we kind of have an idea, but don't really. With phages, you see incredible mutability. If you have a bacteria that evolves resistance to a phage, the phage will develop resistance to that resistance, and so on and so forth. And viruses have been beating bacteria at this race for billions of years. So with phages, you finally have a research platform, but also a modality that can continue to provide protection against infectious bacteria for the foreseeable future. And that's where I think the power of phage approaches really lies.
So just to jump in, for those in our audience that may not be familiar with what a phage actually is, these are viruses that infect bacteria. And I think we're all aware of how powerful a virus that infects us can be in the environment. And this is basically phages, bacteriophage, are using the viruses that infect the bacteria. Larry, you're also in the microbiome space. What are some other strategies that are being adapted to this post-antibiotic battle with our microbiome?
Let me present a little bit of a different perspective here. So all of this is rooted in this germ theory of disease, where there's a bad actor and it attacks us and we have to kill it to get rid of it. But what if that is the final step in a series of events with a number of precursors where basically we lost the resilience? What we see is infection. Let me paint a picture of health a little bit different. So we evolved on this planet most of our time as creatures of scarcity and hunter-gatherers, but we were deeply connected to the microbial world we came from. We had a much richer relationship, of which we've lost most of this. And that gave us resilience. They did all kinds of things for us that we're only beginning to appreciate. Hell, we just decided that we had a microbiome recently. And they have been doing this for us forever. So what we see as infection is inflammation. Now imagine, you know, the game Jenga. So if we were a tower of Jenga and each block is a unit of genetic and metabolic capacity or information, if the tower was 300 blocks tall, only one of those would be human. And in our wild type state, we had all of these blocks in and the tower was robust, but we went from being hunter-gatherers and we developed agriculture, and we domesticated animals. And every time we did that, we pulled connections out, the tower still stands, but what it's lost is resilience in response to stress. The past 30 years, inflammatory disease has nearly tripled. And the notion that we are always going to be at this war, we may actually be able to evolve that notion to one where if we rebuild the resilience, things that would have otherwise triggered this inflammatory response between a bacteria and the host wouldn't actually be inflammatory. We are all running around right now with a burden of inflammation, which is generally subclinical. If I ask you, what health means, most of us will say that I'm not sick now, at least that I know about. But we are now seeing chronic disease at an epidemic rate. 6 out of 10 Americans have chronic disease. 4 out of 10 have 2. Children have chronic diseases. This was not a feature of the human animal as a result of the evolutionary optimization. What we're learning with the microbiome is what we're missing, what we lost, decisions that we made that had implications that we did not yet understand. If we're going to treat disease, we go in as we pick a disease and we poke around in the mechanism, we find a way to intervene to stop the progress of that disease. Restoring health, and that is the big opportunity from the microbiome, is we can go put as much of it back as possible. Listen, we're not going to go live as hunter-gatherers again. None of us would survive, despite what those post-apocalyptic movies show us. But what we can figure out, you cannot restore something if you do not know what it looked like before. And there's about 25 to 50,000 of these humans who still have this. And we have had a unique opportunity to build this model and use that model as a reference point for the metabolic contribution, the functional contribution, and do the experiments to see if we can put it back. And if we put this back, look, there will still be infection, but I suspect there'll be a lot less disease from that infection. One other thing I want to touch on briefly is antibiotics. So one of the things we've discovered is these isolated tribes who've never been exposed to antibiotics, their microbiome has resistance genes to antibiotics they've never been exposed to. Now that may sound surprising, but it shouldn't be, because those antibiotics were all directly or indirectly derived from the microbes themselves, from soil microbes, from fungus, things like this. And maybe they weren't really antibiotics. Maybe what they were the complex molecules that these ecosystems use to self-regulate. What we did is we isolated them and we turned them up to 11, to take a line from Spinal Tap. And in doing so, it activates all of the compensatory mechanisms that are built into those microbes. The place we get into trouble is now there's horizontal gene transfer of that resistance, because we force those genes to be expressed, we increase the likelihood they're going to share those genes, and then bacteria that never had them before now suddenly have resistance. We had a defective model. If we change the model, maybe we don't have to continue to trigger those things in a way that, look, biology's been doing this, microbes have been doing this for billions of years. We're newcomers.
That is a really, really interesting perspective. And I think it's said that our bodies are 10 to 1 bacteria cells to human cells, and coming into better synergy with our microbes can bring us better health. Turning to the other 10% is the cell therapies. This is what Nogami-san is working on. And these have some really interesting opportunities where we can restore or replace function. Nogami-san, where do you see some of the most interesting opportunities for cell therapy in addition to the cardiovascular area that you're working on?
Sure. So one big commonality maybe I can see here will be micro, not the microbiome, but we call it microenvironment of the organs. In at least for the heart failure, many people have been trying to replace the bad cells with the healthy cells, so that people believe the organs should be healthier. But it tends to be the heart failure is a state of the heart that cannot actually recover by itself, and even replacing the cardiomyocytes or heart muscle cells doesn't lead to the better cardiac functions. Before that, we have to modify the microenvironment of the heart so that the heart can accept the newly introduced cardiomyocytes, because heart is protecting itself from the heart failure by replacing cardiomyocytes with fibroblasts so that the tissue can be more stronger and stiff. But and then when it comes to the chronic organ diseases, we see an opportunity of changing the microenvironment by way of supplementing not the main cells in the organ. So like the main cells I call here is heart muscle cells or cardiomyocytes for the heart, and maybe not for the other organs. But apparently fibroblast has not been thought as a functional cells. It doesn't like pump out the blood. It doesn't move. So until recently, nobody knew that what the fibroblasts are doing, and basically the cell therapies are trying to eliminate the fibroblast in the cell culture of the cardiomyocytes. But we found that the microenvironment in the organs is actually really important for having really good recovery of the organ functions. So we see clear opportunity for the other organs as well, for modifying the microenvironment to get the organs have the natural course of the recovery.
And in your introduction, you mentioned that you are about to initiate clinical trials. That is a fantastic advance. I know how hard it's been and how amazing you and your team have done in taking the technology there. Cell therapies and all of these living therapeutics are really a very new type of therapeutic intervention. What are some of the challenges that you saw, or some of the challenges you see ahead for regulatory bodies really understanding and advancing these therapies in a robust manner?
So one of the challenges that the cell therapy companies are facing is immune system in the body. So from the regulatory perspective, it is unwanted to use the additional medications, especially the immunosuppressants in a clinical stage. It is apparent that we have a lot of side effects from using immunosuppressants, and there has been a lot of discussions in Japan as well if this is acceptable to have such a severe medications along with the cell therapies. But now some of the companies figured out the ways to use them, or autologous treatments are becoming a clinically viable option. And we just some days ago, we have seen fourth approval for the CAR T treatment from Bristol Myers Squibb, approved by FDA. And we think that the autologous has not been thought as a clinically viable option due to the expected cost. But seeing the efficacy with the autologous cells and knowing the side effects arising from the immunosuppressants, it seems to be a clinically viable option as the manufacturing cost is reducing as well. So yeah, but still, how we tackle this immune system is a huge issue for most of the cell therapy companies, and that is having the regulatory perspective and the cost, and the autologous and allogeneic discussion as well.
Awesome. So feel like we've covered a lot so far, with starting from like astrophysics and more phages than stars in the universe and 10 times more phages than bacteria, to getting really specific on applications, like replacing like heart replacements, and how do we make those effective. I know, Yug, you said something about the clinical trials and work that your group is doing. Maybe I'd love to hear more about like what specific applications are Living Therapeutics addressing today. And maybe from there we can launch into what might happen in the next 10 years or more.
Yeah, I think it's really interesting to understand where we stand in the field of microbiome research, right? I think at this point it is clear to us, and is becoming even clearer, that the microbiome impacts all sorts of aspects of health and diseases in our body. Everything from skin diseases, gut dysbiosis, Crohn's disease, IBD, Clostridium difficile infections, neurological disorders. There's implication in Alzheimer's, Parkinson's. There's even autism implications. And as we learn more and more about these, or the impact of the microbiome on these diseases, we're going to realize that there are probably some good bacteria or good strains that promote health, and there are some bad bacteria or bad strains that tilts the balance more towards dysbiosis and that need to be eliminated. The first task will be to ascribe a causal link to them. So in many diseases, we now say, oh, in a disease state, we find bacteria X. In a healthy person, we don't find bacteria X. But can we link bacteria X to the disease? That is a more interesting and more useful question. Phages can actually help do that, because you can literally remove or delete species X from the community and then say, oh, does the sick person get better? And I think that as we learn more and more about the microbiome, this ability to subtract as a tool is going to be more and more important. And I'll just give you an example.
We don't have it yet. Does this exist yet?
Yes, and I'll tell you right about it. So with acne, we've been doing this. And in acne and also in eczema, there are bacteria that are central to the disease. In acne, it's C. acnes. In eczema, it's Staph aureus, that have been correlated with disease. And there are decades and decades of literature that establish a very strong correlation link. But the causal link is missing. So what we've been able to do with clinical data now is show that first, when you apply the phage that targets the C. acnes, can we reduce the amount of C. acnes on the skin? The answer to that is yes. The second question is, does that decrease in C. acnes correlate to an improvement in acne? The answer to that is also yes. And the really interesting thing here is, in our group where everyone was given phage, there are some responders and some non-responders. In other words, some people's acne gets better, some doesn't. The really interesting thing here is, and comparing apple to apple, so everyone's given phage, the people whose acne got better exhibit a much sharper decrease in C. acnes than the people who don't respond to the treatment. And that, for the first time, is establishing the start, I would say, of a causal link between C. acnes and acne. And the last question we asked was, what is the effect of decreasing the C. acnes on the skin, right? How does it affect the microbiome? What we observe is that when you decrease the amount of C. acnes, which is very, very dominant in your skin microbiome, it allows the other native bacteria, the good bacteria that don't cause inflammation, to bloom, and it increases skin diversity overall. We use a metric called the Simpson's Diversity Index. And to Larry's point, there have been many observations, both of the Yanomami population in the Amazon, but around the world, where they say that people with acne have lower diversity. And if we can just increase their microbiome diversity, we can maybe shift them to health. And that there are certain ancestral populations where skin diseases don't exist, which have incredibly high diversity. And so we're seeing here, using phages, that we can shift people from a region of low diversity to a region of high diversity. And maybe that is a way, if we can sustain that high diversity, to get them to have clear skin for life, instead of that familiar cycle of getting better, then a relapse, then getting better. And so this is the start, I think, we're just sort of dipping our toes and dangling our feet off of the edge here, but there's a very deep ocean to explore.
Can I dive in off that and maybe tie something that Nogami-san said to what you were talking about, Larry, and ask you a question here? He mentioned that with cell therapies, there's no need for immunosuppression. That maybe brings together some synergies between these modalities. I know you've been in this industry for quite some time. Could you opine a little on that and maybe give us a prediction of where you see some of these things going? Are there medical interventions or things that are done today that you feel will become obsolete by the innovations happening across these live therapeutics?
I hope for obsolescence. What I would aspire to at this point is a significant reduction. And I think what we're finding here, and this is all very new, and a lot of this stuff we're putting together into a manuscript, so I'll share the part that I can. The skin microbiome actually is more complex than we appreciated. The problem is that it's only been studied on Westerners in the kind of depth that we have. And it turns out we're missing a great big piece of it. So the skin microbiome that we see when we swab a Westerner, that I'll refer to that as the epidermal microbiome, and that's engrafted into your tissue. I mean, you could run alcohol over it, and we do, and it's going to grow back. And by the time you reach adolescence, that's pretty much locked in. And there's a thing called colonization resistance. Just think of it as your first line of defense. This is what it's supposed to do. It has, while you were very young, from the time you were born until it gets locked in, it has conditioned your immune system. So they have a friendly relationship. The second thing is that it is deep and it is locked down. So any newcomers that might want to show up and get foothold, there's no room at the end. It's also just as unlikely that you'll ever kick anybody out. Now, the relative abundance, which is what Yug was talking about, can shift. So, if someone's there, it could end up with a little tiny piece of real estate or a big piece of real estate, depending on your skin conditions. But what we don't have that they do is they have a much richer layer sitting on top of that, and that is in dynamic equilibrium with the world in which they live, with the river, with the plants, with everything. And that is sort of an ecodynamic layer of your microbiome, and it's much richer, and it changes all the time. You can go from one environment to another. What we have learned is all it takes is one shower with soap and it's gone. It's a rather fragile biofilm. It manufactures all sorts of important biomolecules, vitamins A, D, E, K. It creates a layer that protects us from UV radiation. They don't get sunburned at all. Yes, they have more melanin than I do, but they can stay out in the sun indefinitely. And all animals have the same thing. It's been studied in animals as well. So we have these complex ecosystems that have enveloped us from the time we ever existed as a species. And everyone had, and in the past certain period of time, a couple hundred years, we made decisions that depleted it and then removed it. And this has important implications for who we are and the way we see ourselves as a species. You see, we were told that we were apart from nature and biology, not a part of. And we're suffering from a bit of an existential identity crisis. Now, suddenly the blinders have been taken off. We have these technologies that we can suddenly look into it. And what we're discovering is our immune system has been conditioned early in life to live in harmony with whatever that set is. And as things get introduced subsequently, it's likely that it will trigger, again, inflammation. I'm going to say this over and over again. You'll hear inflammation from me so many times, you'll get tired of it. But in fact, that is the final step. That is the clinical manifestation of a system that has failed, that has gone from being resilient, where it bends, to being brittle, where it breaks. And so, as with the cells that we're putting back into the heart, we already have a relationship with them. They are autologous cells, if I understand correctly. So our immune system recognizes them. With the types of interventions that we're looking at, I know that we're not going to grow this layer back on our skin. One, no one's giving up soap. And two, we live in a built environment. The layer that we would build is not the one that was part of our legacy. So what we've done is we've identified its metabolic, essentially systems biology contribution, and we have rebuilt that. And we're continuing to rebuild that. Look, as Yug said, we're standing on the very shore of a very deep ocean. Everything that we think we know today, if we're lucky, some of it will be a gross oversimplification of something beyond our imagination. And a lot of it's going to be wrong, but we are going to keep evolving it. And what's happened now is suddenly, most of the life on this planet that was invisible to us is visible. I want to make one quick correction, Caleb, and then I'll shut up, which is the previous notion was it was a 10-to-1 relationship in terms of the number of cells. That it keeps getting revised. Now people are saying 1-to-1, but I think it's the wrong perspective. It's the amount of genetic capability that they have. It's at least, we have about 25,000 genes. Our microbiome has 300 times that. Our genes are relatively new. Animals only came into the picture 600-700 million years ago. Some of these bacteria have been at it for billions of years, and they're good at it. And if we get really, really smart and really, really clever, maybe someday we will think like a bacteria.
They are further evolved than us in many respects.
By every respect.
Larry, a lot of what you're mentioning to me reminds me of a concept that I learned from a bunch of my time in Japan, which is Mibyo, a different state of disease. And rather than just having healthy and sick, there is this intermediate state in Japan called Mibyo, and treating that. And Nogami-san, based on what you've heard from some of the others, do you think that Japan or the Asian ecosystem is better equipped for adoption of cell and gene therapies? I mean, with cell therapy, there's obviously Yamanaka sensei and an astronomical amount of brain trust and research, but do you think there are some cultural attributes and ways that the other living therapeutics may be more rapidly adopted?
Well, yeah, the connection, like the culture, I'm gonna start from explaining Mibyo a bit more. Mibyo is we reminded Mibyo is kind of new idea to us as well. Actually, we used to have it 1,000 years ago, and then we get this idea again in the years of 2000, maybe 1918. This is not distinguishing the sickness and the health. So this is the people feeling something is bad, but that is not clear what is wrong with that. Or we got some diagnosis. This is not a disease yet, but this could be some time could be a disease. So that is a state of Mibyo, not yet sick. And then the people are trying to use this idea for promoting diagnosis and promoting the new modalities to avoid or prevent being sick. So, and then from the cultural background, I'm quite not sure. People think we love probiotics, and the Asian culture have a bit of different perspective to the microbiomes, and I think we prefer to use naturally originated medications, even if that efficacy has not proven in a clinical trial. The people with the thousands of years of experience using such natural materials, we know that it's good for the health, and that could lead to treating the Mibyo status of the human. So I think there's clear fit between Mibyo idea and the living therapeutics for avoiding and preventing the sickness.
And the regulatory system in Japan was updated to facilitate cell therapies. Could you explain a little bit of that to our panelists?
Sure. So we got a new regulations for the cell and gene therapy people know that this is extremely hard for treating and doing the clinical trial for the chronic diseases. We may need to have really long clinical trial period and having thousands of people recruited to the clinical trial. For the heart failure, this is not surprising to have more than 50,000 people recruited to the clinical trial. Then the none of the biobenches would be financially equipped for doing such clinical trial. So we got a new regulations in 2016, I believe, for the conditional and time limiting approval for the cell and gene therapy products. So that we have seen one of the products for the heart failure, which is called heart seat, a seat type product attached to the surface of the heart, which helps heart to pumping out the blood again. And that has been approved with only 7 patients in a phase 1 clinical trial, observed 1 year from the attachment of the seat. And the approval is for 7 years, and they need to report all the treatment results after the approval. Then that product will be going to have the review for the post-marketing survey, and then that could be lead to the final approval. So it is really beneficial for all the cell and gene therapy companies doing the Japanese clinical trial, so that they can have early sales for selling the product as a approved product. And once the product is approved in Japan, then that automatically means the product can have the reimbursement from the government-backed insurance. So this is promising that once you can have the efficacy and the safety kind of confirmed in Japan with a limited number of patients, then the product will be going to be reimbursed from the government.
Okay. Thank you. That was a really good summary of one of the critical challenges, it sounds like, in making all of these wonderful approaches that now we know about and people are learning about possible, is that regulatory challenge. Are there other things that we should be thinking about? You know, it sounds like this is all great to go. Now we understand bacteria make us healthy. It'll reduce inflammation. The phages will take out the bad bacteria and we can restore our heart cells and everything will be great. And clearly it's not that easy. So I'm curious, perhaps Yug, you could comment on what are some of the manufacturing and other, non-regulatory challenges that we still need to overcome for these therapies to be successful?
Yeah, so we've been in the skin microbiome space for a while. And one of the big hurdles is manufacturing, because for phages, I'll speak for phages, but even for, a live bacteria, some of these bacteria are anaerobic, slow growing, are non-canonical. So they're not your garden variety E. coli, and they take a considerable amount of expertise to make. I remember having a conversation with Larry many, many years ago where he was telling us what he had learned from his previous company. And he said that, sometimes you realize that you are the expert in the whole world of growing this kind of cell just because you've been doing R&D for a long time with it. And we took that to heart, and it was very difficult, but we built our phage manufacturing from the ground up within our company instead of outsourcing it, because ultimately what happens sometimes is that other people learn on your dime, and then you need to have that that knowledge in-house. What that builds for us is also a technological barrier that other people cannot surmount. The other thing is, and so for manufacturing, in short, I'll say that I think when you're kind of on the edge, if you can't find an expert to manufacture it who has proven track record to do it, you should probably try and do it yourself. It'll be difficult, but probably well worth it. On the formulation side, this is something that we've worked on as well. Phages are technically non-living because they're not metabolically active. You could say the same to lyophilized bacteria, but those are still considered living, although in a suspended state. So I prefer to think of replication competent phages as alive. And as alive entities, it is actually quite difficult and a considerable challenge to formulate these into skincare, because pretty much all skincare has antimicrobial preservatives that are supposed to kill living things. So we were able to actually solve that problem. It's not a trivial problem, and it required a lot of engineering, and we basically had to build a formulation unit again in-house. And so these are some of the challenges. I mean, there are some regulatory challenges as well, and I'm sure Larry can probably talk about it in a much more informed way. But that's, I think, part of the puzzle as well.
Thank you. And we got a couple questions coming in from the audience, and I welcome the audience to add more questions to the chat. Please pepper us with all your insightful questions. But diving off from Yug's point and over to you, Larry, the traditional biopharma, you want to take a sterile product, a sterile pill, a sterile injection, and for all these living therapeutics, sterile is pretty much worthless for a live biotherapeutic. Do you see changes in manufacturing, or what are the things that, as you're building your company forward, you really worry about?
So this is really a transformational science, and it's going to challenge every single one of the institutional and regulatory pieces that touch it. So there's going to be some real challenges in intellectual property around these kinds of things as they go forward. The stuff that we saw with genomics before is a shadow of what we're going to see as we get into these living interventions. Similarly, the regulatory side of this, and as Yug said, I'm pretty sure I was the first person to put a living bacteria into a cosmetic product. And what I will tell you, Carly, is I don't plan on doing that again. It is immensely challenging, and also, by the way, generally not necessary to do so, is what we are learning. But at each point in the way, it's going to cause us to rethink institutions and standards and practices that evolved in an earlier era. And the way I'd like to think about this is the life sciences, as a result of what we're doing today, is very much where physics was 100 years ago. And you can imagine these Newtonians watching this oncoming revolution and dismissing it as either irrelevant or wrong. And it wasn't that what they were doing with Newtonian physics was wrong. It was just a small piece of a much larger, much more dimensional universe that they couldn't at that point imagine. And the people who will transform the field that you're working in tomorrow don't work in your field today. So it's not as though you're exposed to a lot of these things. And the problem with being the leader or the visionary, the forefront of this is, you're swimming upstream the whole way. It is also the most exciting and exhilarating place to be. And having sort of played at that interface, I can see where we're going to be. One of the questions you talked about asking is, what would someone 10 years from now be surprised? Okay, here's the pathologically optimistic part of my pathologically optimistic cynicism, which is we will be surprised how easy it was to recover our resilience and not have as much disease to treat. There's a staggering observation that was made in one group that went down and did non-contrast coronary artery tomography on 705 Bolivian hunter-gatherers. I have no idea how you do that, by the way. And what they discovered is in their 70s and 80s, their coronaries had lower calcification scores than ours do in our teens. And they didn't have any other diseases. They may not even have cancer, by the way. But
wait, Larry, so this is, they were going through the dead bodies and looking at the tissues and seeing what diseases
no.
Is that what?
No, they brought the instrumentation to do tomography on their non-contrast coronary artery tomography, where you get a calcification score of your coronary arteries. And they did that in the jungle on 705 hunter-gatherers. It's a major accomplishment. Here's the remarkable observation. We have a biomarker, we call it C-reactive protein, that we use as an index of systemic inflammation. 50% of those people had biomarkers over 3, over what we would call inflamed. We would call them inflamed, but they were not inflamed. And what this tells us is we don't know what a healthy human looks like because we've never met one. And once we get that window into this, we will suddenly have tools and capability so that, yes, it's important that we develop ways of treating disease because there's a huge burden of it out there. But wouldn't it be amazing if we discovered there were relatively simple things that we can do that we didn't know that we were doing wrong? Restore the resilience and have less disease to treat. And when we do that, and as we do that, regulatory, intellectual property, business, we're going to have drugs that make copies of themselves. All of these things are going to have to rethink a lot of their deeply held beliefs. We are suffering from hardening of the categories as well as hardening of the arteries. And both of those things are going to surrender to a new paradigm. In the movie version of my life.
We have drugs that make copies of themselves today. mRNA vaccines, right?
We do.
That is here today.
Yep.
And maybe a couple of years ago dreamed far off. What you said reminded me of the saying that innovation by definition comes in the face of conventional wisdom. And what you're saying is that we are at such a precipice moment in changing the way we deliver therapies that these challenges are all massive opportunities for retooling the way we deliver and think about healthcare. And for all of our LPs and investors in the audience, it is an honor and a privilege at Prime Movers Lab to be sitting there and looking at those. And I also thank you, Larry, for seeding the question that I want to add to Ken and then Yug. What in 10 years do you believe will be commonplace that people doubt can happen today? Nogami-san, please.
Sure. I don't know how many people here are believing in a personalized medicine is going to be true in 10 years, but I strongly think that the personalized medicines, and especially from the introducing new diagnostic modalities and figuring out what is the core root cause of the disease, is leading to finding the optimal treatment for each of the patients. So as we introduced 3D mapping system to the heart failure diagnostics, all the other new diagnosis equipment will lead to the personalized medicine. Today's diagnosis is still really broad, and it is almost impossible to choose the right cells or right genes or any type of new modalities, which is going to be suited for each of the disease. The heart failure is the typical. We call all the infunctional heart as a heart failure, but there's tons of the cause behind the patients getting their heart failure. So we think that within 10 years, the actual clinical practice will be accepting the new modalities, and there should be tons of evidence behind the new diagnostics. And then we should be able to see more cell therapies, gene therapies, targeting the specific root cause of the disease, and that can be treated as a personalized medicine.
Yug, same question to you, quick, and then we have one coming in from the audience. We're coming up to the end of the hour. Please put in any burning questions you have. There's an amazing brain trust here.
Yeah, I think that we're already on our way to do this, and the next 10 years, our eyes will be opened even more. But the biggest thing that we'll learn is how the microbiome really affects our body. I'll give you a really quick example. Again, going back to phages, we're basically covered in phages on the inside and out, where, to use a metaphor that as lunch is approaching, we are panko and crusted katsus, and the panko is phage. There was a paper recently that said that there's a non-host derived type of immunity that phages provide us. And this is how it works. In our gut, our gut is sort of like a tube, right? In the middle is the stuff that passes through, and right on the edge is the cells that make up this tube. In the middle is a layer of mucus that kind of coats our cells, and it provides a barrier between the bacteria that live in the center of the tube and our cells, that epithelial cells that line the tube. Now, this mucus, it turns out, is an unusually rich minefield, and the mines are phages. The bacteriophages are incredibly enriched to a rate of 70 times what you found in nature. And what is thought to be the case is that these phages are repellents, so that bacteria trying to burrow into the mucus and trying to infect or make contact with our own epithelial cell will more likely than not find a phage that kills them and just blow up. And so phages provide this layer of immunity to us, which we didn't know existed until probably less than 5 years ago. And they're doing it all the time, and they're probably one of the more potent ways that we avoid getting infected in our gut, because as we all know, the burden of bacteria in our gut is immense, and a lot of them are not friendly. And so just uncovering these new ways in which the microbiome interacts with our body, I think is going to be fascinating. And it is my hope that some of these discoveries make their way, and innovations and insights make their way into products. And that's sort of the goal of our company and many other companies here as well.
Thank you. Thank you very much. We have just a couple minutes left here. Larry, I see you typing the answer that came in from the audience. Thank you very much. Please complete that. I'd like to end on just one quick question to each of you, 1 minute each. If you had a magic wand and could have one technology today, you could just make that technology appear today to help your business advance, what would it be? And Yug, why don't we start with you on that, and then go to Ken and finally Larry.
I would say better sequencing, but I'll cheat and do two. I think engineering phages, which is a technology that's pretty much here. So those are my two.
Kenichi.
Sure. I think the manufacturing cost for the cell therapy is ridiculous right now. We do not have standard manufacturing, so all the manufacturing related technologies will mean a lot to us. And we have seen the CAR T therapies approved more than 300,000 $. So yeah, definitely the manufacturing technologies will change the stage of the business.
Thank you. And Larry, last but definitely not least, can you take us away? If you had a magic wand, what technology would you get today?
It wouldn't be a technology. It would be more inspired, dedicated researchers, individuals who are curious, who are willing to take risks and explore this. The technologies will keep getting better. Our sequencing will get better, or all those things will improve. But what we really need is the human factor in this. Science is a disciplined practice of wonder. Wonder is fueled by humility. I want those people. I'm older. I want them here. I want them to carry this on past when I can. That's what I would wish for with my magic wand.
Thank you all. This has been a wonderful discussion. I thank you again, our panelists, for taking the time, especially you, Ken, calling in at the wee hours of the morning. And I hope you all learned as much as I did. And looking forward to seeing everybody in 2 weeks, same time, same place. Everyone have a great afternoon.
Thanks, everyone. Bye.
Thank you.